My name is Weyand, and I've been affiliated with ITDI since 2015 when I came to Ohio University. So for current projects in my lab, we have two big areas of research. One of the fields I work in is called cellular microbiology. So we're studying how bacteria interacts with human cells, and we're curious to know the mechanisms of infection and how infection changes the infected human cell. So it's an interface between bacteriology and cell biology. My lab is working on Neisseria gonorrhoeae, and this is a pathogen that is really a big problem in the world right now because of antimicrobial resistance. So we're fearful that we won't be able to treat gonorrhea infections in the future. So one of the aspects of Neisseria gonorrhoeae infection we study is called immune evasion. Neisseria gonorrhoeae has several tricks for evading or getting around the immune defenses our body has. And so we have identified some human proteins that Neisseria gonorrhoeae steals from the infected cell, and it uses these proteins to protect the bacterial membranes from damage by immune defenses of our body. Another area we work on is studying aspects of asymptomatic colonization. So Neisseria gonorrhoeae is a human-specific pathogen, and that makes it challenging to study. It only infects humans. You can't put it into laboratory animals, but it will only persist for a short time. So we have taken an alternative approach to study Neisseria species from animals. So one example is we found a new species of Neisseria in wild house mice, and now we put that into laboratory mice, and it will persist for a really long time. And it doesn't cause disease in mice, but it mimics behavior of Neisseria gonorrhoeae in humans, and that is asymptomatic colonization. So many people that are infected with Neisseria gonorrhoeae don't know they are infected with it, and this promotes dissemination of Neisseria gonorrhoeae in human populations. So we're trying to study factors that are shared with animal Neisseria with the pathogenic species in the hope that we can identify and study common mechanisms of persistence in a mammalian host. My students and I try to culture new isolates of Neisseria from a wide range of animals, and most of these isolates don't cause disease in animals, but we're interested in doing comparative studies between Neisseria species that cause disease and those that don't, so we can understand different and shared mechanisms of association with a host. Students in my lab are working with isolates from mice and monkeys, and we enjoy researching just how these bacteria persist in their host and do so in the face of a healthy immune system. So in regards to students in my laboratory, I currently have 4 graduate students that are working towards their PhDs. And then I have 2 undergraduate volunteers in the laboratory that are developing projects. So regarding collaborative work at ITDI, we have a good group of biologists that work on various microbes. And this is a really fun environment to work in and try to help each other as we study different principles of how microbes work, how they're resistant to antibiotics, or how they cause disease. So some of the inspiration behind the research we do is just a basic desire to understand how life works. And so I like to tell my friends that I think small life is beautiful. And I enjoy learning about nature of macroscopic organisms too, but I'm working in a field where I'm studying the principles of life in the context of microbiology at a very small scale, and just interested in understanding how bacteria and their hosts get along, or how they don't get along in the context of infection.